Good afternoon, and welcome to the OR Today webinar series. We're excited to have over 480 registered attendees for today's webinar. I want to remind everyone before we get started that this webinar is eligible for one continuing education hour. The post-webinar survey and the certificate process is now automated. So in an hour's time following today's presentation, you'll receive an email that will have a link to the survey. Once you finish the survey, you'll be able to download your certificate from your desktop or your laptop. Let's get started with a trivia question today. I want to give someone an OR Today Live surprise pack. We have a question this week that is, what's the name of the American author and Civil War nurse who wrote the novel Little Women? Again, what's the name of the American author and Civil War nurse who wrote the novel Little Women? You can answer now using the questions feature on your dashboard. While you're busy answering the question to see if you're the winner of our OR Today Live prize pack, there are a couple of other reminders that I want to share with you. While you're answering, I do want to remind you that if you've missed any of the previous webinars, you can watch them online at ortoday.com/webinars. You can still receive one continuing education credit for any webinar that has happened in the past 12 months. Also, I'm hoping you'll all help us celebrate the Year of the Nurse by sharing a standout story about you or a special nurse in your life that helped you get started on your career path. Each submission will be entered to win a $25 Bath & Body Works gift card. And to enter or find out more details, you can visit ortoday.com/contest. Of course, just a reminder that our 7th annual OR Today Live Surgical Conference will be taking place this year, August 16th through 18th at the Omni Interlaken Hotel in Denver, Colorado. Join us to discover new opportunities, broaden your knowledge and exchange ideas. Yes, registration is open for this awesome conference. You can get more information by visiting ortodaylive.com. All right, let's find out who our winner is. Again, our trivia question this week was, who was the Civil War nurse and American author who wrote Little Women? And Jessica Kim, you're our winner this week. Congratulations. You got the correct answer, Louisa May Alcott. Jessica, we'll reach out to you shortly to get your mailing information. But first, OR Today would like to thank our sponsor, oneSOURCE document site. The oneSOURCE document site includes the most comprehensive online collection of IFUs and service manuals. 5 databases available covering surgical instruments and equipment, tissue and implants, dental, biomed, and facility maintenance. For more information, you can visit onesourcedocs.com. Again, that is onesourcedocs.com. Our presenter today is David Jagrosse, President of David Jagrosse Consulting and consultant to oneSOURCE document site. David, you may begin whenever you're ready.
Great, thank you. Thank you for the intro. And I'd like to thank OR Today and oneSOURCE for allowing me to get in front of such a great audience. And more importantly, I want to thank everybody for getting on the webinar today. And particularly thank you for everything you're doing out there on the front lines. I don't have a bio slide up here in our presentation, but I'll tell you a little bit about myself. I've been in sterile processing for 30 years now, 30 plus years. I came out of a hospital last year, working full time running an SPD. So I'm doing the consulting side of things now. But this is kind of the first crises, if you will, where I'm not on the front lines and in the trenches with you guys. So hopefully I can do my little part by keeping education going and being a resource for everybody on the peripheral. So anytime you need help with anything, always use our networks, reach out, and don't ever hesitate to ask questions or look for answers. I always like to tell people the wheel has already been invented. We always just look for a way to make it go faster. Like I mentioned, I got 30 years in sterile processing. I was involved in my local chapter of sterile processing for a number of years since the 90s and still involved today. I was on the board of ISHM and president of ISHM, our international organization. And I'm still a member of AAMI ST79 work group, which develops our guidelines. I'm no longer a user on that committee. I'm an independent consultant, but nonetheless, we'll talk a little bit about that as we get into the slides, because I always like to bang the drum on getting people involved, because I believe our fate is in our own hands. If we are the experts, we should be developing the guidelines. So, and if we don't, then they will be developed for us, and we really can't complain when we see a crummy standard or something that we're like, hey, this is impossible to follow. So let's jump into the presentation here, and we'll go over our objectives on our slide here. We're going to look at the issues that are involved around navigating multiple IFUs. And even if you say to yourself, well, what are multiple IFUs? We'll cover that. We're going to show you some examples of that. We'll look at a real world example of what could happen when we start trying to make those decisions, and then some of the pathways on how to resolve them. And towards the back end of the presentation, I'll give you some of my Dave Jagrosse input and experiences, and where I maybe kind of see where this is headed, and how we can make it a little easier for the end users. So on the next slide, we're going to ask the question, why are the IFUs so important? And that may be obvious to a lot of people, but it also may not be obvious to a lot of people. I think when you tend to be in the business, whether you're a nurse or a sterile processing person, for a long period of time, you make a lot of assumptions. You assume that people know a lot of things that maybe are commonplace to you. So we got to make sure that we don't make those assumptions. Some people are new to the business. Some people are maybe on this webinar are only greenhorns, and maybe only a couple months into the profession. So IFUs provide the instructions that the device manufacturer states are needed so that we could properly process their device. And this is critical because it's up to the manufacturer. They're the ones who develop the device, and therefore they are the ones best suited to tell us how to reprocess it. Patient safety is a big issue. This is directly tied to the instruments. We want to make sure that we have safe devices. Improperly processed instruments have been linked to patient injury, and not following the IFUs can also put patients at risk. If I jump over to the next slide, continue with why they're important. We have the FDA, we have AAMI guidelines, we have AORN as well. And any professional organization, if you're over in the endoscopy suite, we have the SG&A. So we always look to our professional organizations, and they're always instructing us to follow the IFU, the instructions for use. The instructions for use are actually developed as part of a process that a manufacturer goes through. And that process, in order to sell a medical device in the U.S. market, a manufacturer must obtain what's called a 510K from the FDA. That's the process in which they submit their data so that the device can be cleared for use in the market. And that process typically looks like this. It is a process in which the medical device is sent to a third-party test lab. Some of the manufacturers are large enough, like a Medtronic or others that are large enough. They may have internal labs, but I would say typically 90% of the time they're using independent test labs to send out their medical device. The labs are doing cleanability. They're looking at how to clean the device properly. They're looking at processing it. Then they're looking at the sterilization side of it. That's all included in that very detailed data. That data is then sent along with from the manufacturer to the FDA, where it is reviewed. And if it meets the criteria that's laid out, it will be cleared, hopefully, for the U.S. market. So this is really where our IFUs are deriving from. It's in that process where they're doing the cleaning, they're doing the sterilization, and they're passing that on to us to say, this is what we tested, this is how we tested it, and they're giving us those instructions. Now, it is interesting to note that within that 510K process, that's really where we have a lot of sway. I can give you a little enlightenment about how the AAMI committees work. On that AAMI committee, there are about 150 people that are members or voting members. Most of it is industry, but there is a smaller percentage of people formerly like myself. I'm now an independent consultant on there. There's about 3 or 4 people on there like that, but most of the other folks are called users, the folks that are actually in the hospitals doing the jobs. So it's a small minority. I'd say there's about 20 to 25 people. And, out of that 25, not everybody may be active, but it's super important because that's where we can say things like a number of years back, we said these trays are getting too heavy. So, hey, FDA, when you do this 510K process, when a manufacturer asks for a device to be on the market, like an orthopedic set or a hip tray or a knee tray, let's make sure it's not more than 25 pounds, including the container. So that's kind of where we really have sway or the ability to control or enforce things that we want to see happen. So I encourage people to get involved with their associations, whether it's AORN, whether it's SGNA, whether it's the AAMI committees. There is a lot of crossover. There are a lot of AOR members on the AAMI committees and vice versa. So we always try to make sure that things run parallel and they're not really divergent from one another when it comes to the guidelines. But this is just a little bit of insight of where they come from. That, like I mentioned here on the slide, the cleaning, the testing for function, the packaging system, and then again, sterilization. I like to put that bullet up there because in our SPD world, more often than not, what I see occur is if we're lucky enough to obtain an IFU when the device shows up or when we purchase it, we tend to skip through it. Some of these IFUs are large. Some of the IFUs also contain information for the end user on how to use the device. So it literally can be a booklet, a small booklet. Now, within that booklet, like I mentioned, the SPD folks may get that booklet. And what we tend to do is we skip to the back of the page of the booklet because we're looking for the sterilization. In the past, more recent past, it was all about the exposure time. How long do I need to sterilize this device? Typically, in the past, our standard cycles were 4 minutes of exposure, but now we have these extended cycles. So we would kind of jump to the conclusion of the book, if you will, and look for that sterilization time. And what I'm trying to get across today, or at least with this slide, is don't jump to that conclusion because the rest of the book that you're skipping through is that cleaning, is that testing, and is the packaging. Very important. As we say in SPD, if it's not clean, it's not sterile. And cleaning is a very important function. We can never sterilize tissue matter or dirt or debris. So the cleaning is becoming very specific. You'll notice in the market, if you go look at your IFUs, you're starting to see very, very detailed instructions on cleaning, whether the device requires sonication to be put in a sonic, whether the device needs to be taken apart because some devices need to come apart for cleaning. And then I'm starting to see, and you will start to see, actually instructions for your washers, for the mechanical washers that we use. We're starting to see cycle settings show up. So again, my kind of call to arms is get involved with these committees, because if we don't give these manufacturers the guidance, they're going to come up with their own. These independent test labs are going to start putting into the IFUs the cycles that they used. And what we don't want to see is we don't want to see SPD folks in decontam manipulating the cycle settings on a washer the way that we were manipulating them over on the clean side when we were doing sterilization. Because historically, that's what happened. In the past, we didn't have IFUs, and we kind of screamed and yelled at the AAMI committees and said, hey, FDA, we need cleaning and we need sterilization instructions in order to reprocess these items. And we kind of stopped there. We didn't give further guidance as users. So the FDA required it in the 510K process, and the manufacturers went out there and did all their independent testing. And then we had all these crazy cycles all over the place. 4 minutes exposure, 10 minutes exposure. I think the biggest one I had seen in my time before I left last year, the hospital, was 23-minute exposure time. So now, luckily today, at least on the sterilization side, we've given further guidance as users through the committee and said, hey, here's what typical sterilization cycles look like in a hospital. So try to mirror them when you're doing your testing. We don't get involved and give more guidance towards the cleaning side. That can kind of run away from us. But it's very important just to note, like I mentioned, some devices have to be taken apart for cleaning. Some devices remain apart on the assembly side, and then it's up to the operating room to potentially put it back together, and then vice versa. There's some devices that come apart for cleaning, they get put back together on assembly, and then it's sterilized the way that the IFU instructs us to. Just as a word of caution, if you come off this webinar and then go diligently pick into your IFUs, you have to collaborate with your customers. If you discover that there's devices that need to come apart in the IFU, don't just start doing that, SPD. You have to collaborate with the OR. They may need some training. We can't send them up a laparoscopic or a robotic instrument in 100 pieces and they try to put it together in the OR. We kind of have to do some training and give them a little bit of a heads up on that. Healthcare facilities are not equipped to validate. I put that in there because these terms are used loosely very often. Validation is what occurs during the 510K process. We covered that. We spoke about that. The manufacturers are doing that either internally or they're doing it with an independent test lab. I can't exactly say that facilities are not entirely, they're not typically equipped to validate. What we do is we get medical devices into our facilities. We're looking at validated IFUs from a manufacturer that were cleared, and then we're trying to verify that. We do verification. We're trying to mimic to the closest of our ability the cleaning and then the biological testing and chemical indicator testing that's happening over on sterilization. So there are some facilities that can be high-end, and it's not impossible. You can get ISO or some different types of certifications, but it's very intense to be able to validate in a hospital. But those terms are used very loosely in hospitals often. Let's validate it. Well, no, let's verify it. So you're doing verification. So users have to depend on that manufacturer for the validated IFU. On the next slide, we will see what are the requirements of your library. The requirements are very important. Whether you are doing, there's a couple of scenarios here. You can be doing a paper-based system. If you're doing a paper-based system, where is that located? I know my facility, I came from a 300-bed hospital, just about 15 ORs with 3 off-site and a surgical center. We had about 10 4-inch 3-ring binders full of IFUs. And my history or my experience was that Pam, my ACE technician, I assigned Pam to kind of put that library together to make sure that we had an IFU for everything. And then how do we organize that library? But before we get into the organization, let's just make clear this point, that IFUs are not just for medical devices. IFUs cover the equipment we use. So in an SPD world, it's the washers, it's the sonic, it's the cart washer, it's the sterilizer, it's maybe a demagnetizer you have on the other side, instrument insulation testing device. You know, in the OR, we have freezers, we have, you know, we can go through a laundry list of equipment that's in the OR. So it's just important to note, you know, when you're creating your library, how do you want to create it? Do you want to have the library down in Central Sterile has all the medical instruments, and then does Biomed have a book, or is it online in the computer for all the Biomed equipment or all the hard equipment? The operating rooms have to deal with tissues and implants. Those are coming with IFUs, and that all has to be archived, updated, and accessible. So the key to the library is, like I mentioned, I had Pam go through. I said, Pam, just go through this, go through the department, make sure we have everything we need from an A to Z standpoint. And it's just continuous maintenance. You know, you have an IFU. How do you know it's updated? You may have to contact the manufacturer, and they say, oh, well, what's the reference number on the bottom? Oh, well, it says 2018. Oh, well, we have a new one. We'll send it to you. It's 2020. You know, we're pushing manufacturers to update the IFUs as much as possible. So it is, I can say it was a full FTE in my past facility. That was Pam's job. Not that she didn't do instrument sets, but that was pretty much, Pam was always the guru for the IFUs. We have to make sure that they're accessible at all points. Let's think about the processing wheel for a minute. Let's think of point of use in the operating room. From an SPD perspective, it all begins when the case is over, because that's our processing start point, and we're depending on our partners in the OR to do point of use cleaning during the surgery if required, and then pre-cleaning or spraying with an enzyme foam or keeping those instruments moist, removing gross soils, and immediately when the procedure's over and getting that equipment down to us. So there may be specifics that are needed in the operating room or in a procedure suite. We talked about endoscopy earlier. An Olympus scope may say that they want you to irrigate the scope with 500 mLs of water. You're all familiar with bedside cleaning kits that they have in endoscopy suites. That needs to be suctioned through the scope while it's still connected to the unit, which is on the tower in the room. SPD doesn't have that equipment downstairs. So the point is that person may need access to the IFU in the room. Then we have to look at the IFU. Is there any special transportation considerations we have to make? Gets down to our decontam room, and the game is on. Okay, what do we do? Can I submerge it? Can I not submerge it? Am I taking it apart? Am I not taking it apart? Can it go through the washer, or is this a hand clean device? All these specifics are in the IFU, and we haven't even touched the sterilizer yet. So that's to my point earlier about, you know, there's a lot more to the IFUs than just sterilization. So again, we'll need that access in decontam. Now, if you're in a paper-based system, what are you doing? If all those 3-ring binders are out on the assembly side, are you making a copy and handing it to the person in decontam? Because you don't want to hand them the original and then they hand you back one with a bloody fingerprint on it. Anything that goes to decontam is there for decontam. It's contaminated. So all things to consider. Once it comes out and gets processed, then on assembly, we need information. Do I have to test it? Do I test the insulation? Do I have to reassemble it? Do I leave it unassembled? What is the specifics and packaging system? How do I now prepare it to go into what modality of sterilization? Is it in low temp? Is it in steam? All these specifics. Then finally, we get to sterilization. And then it's not over, because if the device had some kind of special consideration for storage, we'd have to look, the IFU would tell us that. So you think of that whole wheel of processing. Now also think of that wheel of processing at my 3 OR surgical center down the road. How do we maintain that? So the decision was made a couple of years ago to, actually it was 2 steps. We went from the paper version of the IFUs over to scanning those and putting those into our digital computer system so that at least there was accessibility. Pam still had to call and make sure that these IFUs were updated and we had them as new devices came in, but at least it was digital. We could share it with the surgical center. The OR can see it, endo can see it. The next phase after that was sort of outsourcing it, if you will. We subscribed to oneSOURCE. And if there were there's one other company, I believe called Bluefin. They only do like a decontam, but and out of fairness, if there were multiple companies out there, I would mention the multiple companies. But oneSOURCE kind of owns those space with IFUs. It's an online resource. It's a library of IFUs that you can access, and then that updating and archiving is all done by them. They assign teams of people on a weekly basis to contact the companies to make sure those IFUs are updated. One other important aspect that I don't want to forget about your library, in case you are on paper or scan that paper into your computer at the hospital, is your organization system. How are you organizing it, even within SPD? Some people divide it and say, hey, here's the ortho stuff, here is the neuro stuff, here's spine. The users, you have to make sure the users understand how to look something up. If they need to look up a Synthes small fragment set, where are they looking? Some facilities do alphabetical A to Z. So consider that. How are you going to organize it? And then share that training. Train the staff and let them know how to look up something in your system. So let's move along. Like we said, I think we covered all the rest of the bullets, and you guys can read. I don't really like to read when we're doing webinars line for line because you guys are out there and probably already read everything. On the next slide, we talked about how do you obtain the current IFU. Couple of important points here that I like to highlight, and one of them being upon new purchase. So when you purchase a new device, Central's usually not the one purchasing anything. So to my friends in the OR, let's back it up before you even buy a device. Let me give you a trick of the trade that Dave learned the hard way. My past facility had a doctor's procedure they were going to do, and internally in the hospital, but not in an OR suite. They purchased a nice fancy scope that they were going to use. And it went through the capital process. It got approved. It came in. They knocked on my door. They said, Dave, we need you to get this ready for a procedure next week. It's the first time I see it. First time I'm aware of it. The device required ETO sterilization. We got rid of ETO in the 90s. So I unfortunately said, I'm sorry, I can't process this device. It wasn't able to be soaked. It wasn't able to be high level. I said, so they ended up returning that scope with a $3,000 restock fee. So the lesson learned, let's not have it happen again. All of us have a capital process to ask for capital equipment. There's typically a form involved, whether it's in paper or whether it's online in the computer. Just add this to your capital request form. Any devices that are being purchased, ask these 2 questions. Does the device require high-level disinfection, or does the device require sterilization? And if those boxes are checked off, then SPD needs to be involved in the decision process because we don't want to pay that $3,000 restocking fee. Infection control might be trying to control all the spread of, chemical disinfectants throughout the facility. Everybody's soaking in an OPA or in a glutaraldehyde. We might be trying to reduce that. Do we want to bring more equipment on board that creates more soaking or high-level disinfection? So all things to be considered. Nice little trick of the trade there. So be careful with your library. Take those things under consideration. And like I said, you can also outsource this. I mentioned oneSOURCE. They have a number of databases that we used to subscribe to. They have surgical instruments. They have a tissue and an implant database. They do a biomed database for all that equipment I talked about. And then they have a facilities database for the general hospital, and then they have dental. So it's not just limited to sterile processing. Okay, on the next slide, this is called Hitting the Crossroads. And, after looking at this slide, I said to myself, just a small thing, but I'm kind of jumping in saying, like, IFUs are a crossroad sometimes. I like to refer to them as a roadmap. IFUs tell us what to do, right? So think about being in your car. It's like a roadmap. Sometimes you're looking for instructions, and the IFU leads you to a dead end. Okay, just like sometimes you ever be driving around using Waze or GPS, and it just kind of leaves you somewhere where you hopefully not in a lake, but it'll just leave you on a dead end. So we want to make sure that we're looking for, they're not the answer to everything. The IFUs we wish can tell us everything we wanted to know, but sometimes they don't. We might want to know, does this device come apart for cleaning and sterilization? It might not say anything, so they consider that like a dead end. Sometimes an IFU will give you the answer. It'll get you right to where you want to go, and that's a great thing. And hopefully it's clear and concise and quick. Another scenario is it leads you to an intersection, and you're at a crossroads because now I have multiple IFUs. And, I'll give away the punchline right now. I'll give away the whole seminar. The answer to anything, to most of what we do, is critical thinking. This is where the critical thinking comes in, and where we have to use our skill sets and our teams that are out there and available to us to get to these conclusions. Anything you do, even outside of IFUs, I always tell people in sterile processing, you never want to make decisions in a vacuum. Always use your teams that are available. Even if it's as simple as an email, what do you think of this? I'm working on a process. I'm working on a standard operating procedure. I'm working on a policy. You get some documentation. You bounced it off of infection control, your OR team, risk. So don't do things in a vacuum. If we jump over to the next slide, this is where I was kind of thinking after looking at this scenario where I said, okay, we have an instrument going inside of a rigid container, which is then going inside of an autoclave. So IFU for the device, IFU for the rigid container. Rigid packaging systems have their own 510K. A medical device does, and so does a sterilizer. So in thinking about this, I said, well, I could have even taken this further back because, like I mentioned earlier, we have cleaning going on. We have washers and sonics and irrigating sonics going on and decontam. But maybe for the interest of time and space on my slide, we'll just jump right into this. So let's look at this scenario. How what would this look like? Because we don't want to make assumptions, and they each have their own instructions for use. So let's jump to the next slide. This is from that happened to be a Getting a 733 model of a sterilizer. And it's telling, and I'll show you this on the next slide. We're not there yet, but this is the cliff notes of the IFU. 20 each. You can put 16 20-pound trays into the sterilizer. I put this up there. Who has 16-pound trays? If Amy tells us we can be at 25 pounds with container, trust me, those things are 24.99 pounds each. And just a note on that, Amy grandfathers things. So I know some of you out there are saying, oh, what about that 42-pounder we still get? In 05, 06, when the 25-pound rule came back in, that was from that date moving forward for approvals. So if your device was approved prior to the 25-pound rule coming into place, it's grandfathered in. But you, as a facility, can say, we, as a facility, do not want trays over 25 pounds. Go back to your Synthes, go back to your J&J, go back to your Depews and say, guys, you need to reconfigure this tray because as a facility, we don't want trays that are 42 pounds. So just a quick note on that. But let's dig a little further. Let's talk about this a little more in depth. Why 20 16-pound trays? An autoclave vessel, by nature, by the way it works, steam comes into the chamber and condensates. The steam transfers its energy into the instruments and collapses back into water. And that's condensate. So the magic, the trick of sterilization is that the entire load is soaking wet with water during this come-up phase, the exposure phase, and then when it's done with the sterilization cycle, we have to re-evaporate that liquid condensate. We have to dry the load. So it's scientifically known on the steam tables that a vessel of this size, this isn't an exact number, but for the sake of an easy visualization, let's say this vessel can create 1 liter. They know scientifically that 20 16-pound trays in that autoclave will generate 1 liter of liquid condensate. They know by their testing that the autoclave is also capable of re-evaporating that amount of liquid condensate. So, lesson being, the minute we put that 1 extra tray on there, now we're generating 1 liter, 10 ounces, let's say, of condensate. Now, the autoclave isn't going to say, the steam table isn't going to say, Dave, you're dealing with COVID. I know you're short-staffed. We're going to give you a break today. The autoclave doesn't care. The steam table doesn't care. It's only capable of re-evaporating that 1 liter of liquid condensate. This can be a cause of wet packs. That condensate needs to go and end up somewhere. So very important, very important. And you see, it's almost like ignorance is bliss. Now you know, like there's these IFUs and like, oh boy, what do we do about this? How do we address it? We'll touch on that, but this is just sort of like the eye opener and explaining why that is. It's not, and it really depends. It'll go up if it's a bigger vessel. It'll go down, obviously, if it's a smaller vessel. Okay, so I'm going to jump to the next slide, and that's simply going to show you the actual IFU. So like I did the Cliff Notes for you. I'm going to jump over to the next slide, and this is from Aesculap. They are a company that makes the rigid container systems, one of many companies out there. And part of the highlighted section up on top, this was interesting because my staff said to me, can we stack rigid containers on our autoclave cycles? And the answer to that is yes. If they're all Aesculaps and you look at the IFU here, they were looking for an answer of how many. Can I stack 1? Can I stack 2? Can I stack 3? Of course, the Aesculap IFU says neither. Stacking should not exceed 16 to 18 inches in height. So I'm getting instructions from the autoclave that's telling me don't put more than 20 16-pounders in there. But then the IFU here is telling me, oh, wow, look, I could stack 16 to 18. Trust me, I see technicians that can fit 40 trays in an autoclave cycle. They are unique. It's like the game Tetris. Like you'll see trays tucked in there sideways if we allow them to. So this is where that balance comes in. This is where that critical thinking comes in. Like, wait a minute. Okay, I know I can stack. Well, stacking might allow me to put 28 trays in there. No, stop, because we read the other IFU and we have to stop at 20 16-pound trays. Let's jump over to the next slide. So under that scenario, now what I didn't put up there was an IFU for the medical device. So if all was, I think that was like an ENT instrument maybe that I had up there on the slide. So out of fairness, let's say that medical device said, this thing requires 4 minutes exposure at a minimum of, sometimes it's very low, a minimum of 10 minutes. Because maybe when they, and we'll talk about this a little later, maybe when that instrument had its 510K and they did their testing, they put it in a peel pouch. Now, if you put it in a blue wrap or you put it in a container, you're going to start getting different dry times. And there's a whole other scenario of circumstances that can change and different variables. In Central Sterile, we try to come up with dry times that are based on experience. I see it varying from as high as 60 minutes on the dry phase to 45 to 30 to 0. And you might say, well, 0, there are rigid containers out there that have 0 dry time on them, like the one tray. So, if someone's doing an entire load of loaner devices in the cycle and there's no other device on there except the one trays, they may be doing it at 0. So again, a whole other territory, whole other realm. We could have a separate webinar on that in alone, but don't ever confuse your sterilization or terminal sterilization with dry time. The dry time is important because on certain devices, we have to make sure that they are dry. Wetness post-cycle, a wet pack, can indicate contamination. We have to assume that the device is contaminated unless it's in a device that's approved for that. And it's not just the one tray. There's some IUSS devices you can use, like a Riley flash pack, where there's moisture. I believe Steris and Case Medical have devices that are similar to that. Wetness inside the container does not always necessarily mean it's contaminated if the IFU indicates that. So over here, I put up addressing the multiple IFU scenario. AAMI has the ST79 group has looked at this, and it's in the document. And over here in the center of the thing says when there's an unresolvable conflict between sterilizer IFU and device IFU, SPD technicians should follow the device IFU. That's always been pretty clear to us. But then they also instruct that in the case of an unresolved conflict regarding cycle parameters between the container and the maintenance system, then the directives are to follow the container system. And if we go to the next slide, this is directly out of the AAMI ST79 2017 document, section 10.2.2 on sterilization cycles. And it says the sterilizer's manufacturer's written IFU should be followed for operation of the sterilizer and indications for use. And then we have this A and B scenario, with A saying differences between the program cycle and cycle parameters recommended by the device manufacturer should be investigated and, if possible, reconciled before the items are sterilized. If there's differing instructions, the device IFU should be followed. So they're giving us guidance here to say, if it's not clear, contact the manufacturer. The manufacturer should be able to resolve that for you, whether they may ask you what kind of autoclave you're using. They may ask you what kind of packaging system you're using. So you might have to give them some more answers to get your answer. And then over here under scenario B, it talks about a rigid container system. So we're looking at a rigid container system, and that's maybe designed for IUSS as a packaging system. The container system manufacturer's written IFU regarding exposure time should be consulted and reconciled with that of the sterilizer manufacturer. So we have two scenarios here where if a device is going into the autoclave, we're saying an IFU of a device and an IFU of an autoclave, call the manufacturer, see if you can get a resolution, but then ultimately go with the device. So we kind of have device versus autoclave. On B, we kind of have rigid container versus an autoclave. So they're giving us guidance there. Now, in the interest of time, you guys can read, and you have these slides in front of you, and hopefully you all have the ST79 document. I'm mentioning here that do we need a C? Because a section C would be, what do we do? How do we resolve placing a medical device into a packaging system, or specifically a rigid container system? because a rigid container system has a 510K as well as the medical device. And I think it would give us a little more clarification if, and I didn't mention this earlier, the ST79 2017 document right now, there are a couple of amendments that will be coming out this year, hopefully. We might have had a little delay like everything with the virus, but we're under a full revision. The document is up and ready for a full revision again. So it's my hopes that we can get a little more guidance on this and maybe get some more into the document. If we skip over to the next slide, this is talking about how the next revision is coming up. There already is another document out there called TIR. Whenever you see that, that's a technical information report. TIR 12 is being updated as well. And, you know, this is great guidance right here. So, like, while this is all happening, in the meantime, AAMI advises SPD teams to conduct a risk assessment to determine how their facility will resolve these situations. They should do side-by-side analysis of the pertinent IFUs as part of the risk analysis for each scenario, and then develop robust policies and procedures utilizing recognized industry guidelines for the following. And you guys can see that all there on the slide below you. So that's important guidance. It's always the end scenario is to have written SOPs or policies and procedures in place. And how did you develop those? Did Dave sit in the office and do it, or did he pull the team together and there was a broad OR risk, infection control? We went through the documents and we all signed off on it to the best of our ability using the best documents that we had available. If we go to the next slide, again, repetitive a little bit. I said, you know, ST79 is under revision. Hopefully we can get what I'm calling the C tab in there, because we had A and B, we had device versus sterilizer, and then we had container versus sterilizer. But give us a little more guidance on, you know, in specifics on medical device inside of a rigid container system. You know, on the next slide, this is redundant. I put the same slide in there twice just to remind everybody that everything is being updated. We're under full revision, and with the unresolved conflicts, what to do is there on the bottom. If I jump ahead, there's the TIR 12 that I spoke about earlier, and a major focus of that revision is aligning it closer to the ISO. We draw a lot of our updates and guidelines out of AAMI from the International Standards Organization, and Ralph Basile is on that group. He's great, runs part of HealthMark. He said it's going to provide a deeper dive into processes used in healthcare facilities. So hopefully that's being worked on. If people are interested in that, I would suggest to get involved in that. If we go to the next slide, again, let's jump in. Here's some of my own 50,000-foot risk assessment recommendations. When it comes to doing these risk assessments, if you pull the team together for sterility maintenance systems, I want you to keep in mind when you're looking at these devices that a lot of people have a lot of trouble with dry times. And for your consideration, I want you to think that or know that when device manufacturers submit their test data during that FDA 510K process, the FDA requires them to use one packaging system. 98% of the time, they use blue wrap because it's affordable, it's readily available, it's almost pretty much the same. There's a couple of brands out there, but they're all to the same specifications. If they choose to use a rigid container, they'd have to, well, which container system are they going to use? If they use the peel pouch, what brand are they going to use? So just know those dry times are a derivative of this testing. And in a blue wrap, we have to make sure that a device is completely dry. So, or else we'd have contamination. And if you're using a system like a one tray that has zero dry, you got to look at their IFUs and their test data, and that's where the team's doing their risk assessment and then using the best information they have available to them to develop their policies and procedures. The IFUs can call for as much as 1 hour. Like I said, there's some trays out there that call for it. I've seen that on the Smith & Nephew loaner trays that are wrapped in blue wrap. And that's because, like I said, you have a lot of metal mass in there and it takes a long time to dry those kits. If I skip to the next slide, I said a good unofficial rule of thumb from my perspective is to default to the medical device for cleaning and sterilization. And then you have to weigh and look at whatever maintenance system you're using. Is it a container? Is it a blue wrap? Is it a peel pouch? Because that'll help you dictate or try to find where your times will be. Each facility should clarify these murky areas for themselves based on all that information. As a member of the AAMI working group, I kind of committing to you guys to try to clarify this a little bit more on the next revision. But please come out there with me. I need your help out here where we need more users on the committee. If I jump to the next slide, I'm going to get on my soapbox for a minute. I want you to get involved, whether it's AORN, whether it's SGNA, whether it's ISHM, whether it's AAMI. Come join what I call the AAMI Army. Let's develop the guidelines, or they're going to be developed for us. And I don't want to hear anyone cry over the spilt milk unless you spilt it. Let's get involved and let's guide these manufacturers because we are the experts. This virus has proven that a lot to us, and I'm trying to keep my peers in SPD confident because online and in conversations with people, I hear the statement, they told us to reprocess this single-use device. Well, why are they telling you? You are the expert. Have confidence in yourself. Be the expert that you are. This, if I jump to the next slide, these were CDC numbers prior to the virus. We had 1.7 million healthcare-associated infections a year, 100,000 deaths in the U.S. a year. 22% are known to be surgical site. So 22,000 people prior to any viruses, COVID-19 viruses, $20 billion a year. Are we doing absolutely everything we can do down to the details that we can to fight this? Because people, I always like to say, these aren't patients. They're not customers. This, God forbid, is us. It's our family members. Could be our kids. So that's how serious I take it. Thomas Paine, if I jump to the next slide, Thomas Paine said during the Revolution, those that expect to reap the blessings of freedom must, like men and women, undergo the fatigue of supporting it. So if you wanted to be free, you had to go and fight the British. You didn't just go home at night. You went home and got a musket. You went home and got a pitchfork, and you fought for what you thought was right. So I'm calling you guys to arms. Get involved in these guidelines and committees, and let's make it the best thing we can. If I jump to the next slide, I like to say that change does not come about through expectation. Don't sit there and say, they need to change. You know what they really need to do? I always like to say, they are we. We are they. You are they. If you want to make it happen, let's make it happen. And with that, I'm going to jump to the next slide. I want to say thank you again to the entire team. And I believe we do have time for some questions. And with that, I want to say thank you to OR Today, the audience, and oneSOURCE. Thank you.
Well, David, thank you. That was a fantastic presentation with a lot of information for the attendees today. And while you were presenting, a couple of questions came in. I know we're coming up close on the 60-minute mark, so let's see how many we can get through. But I want to let attendees know we'll send all questions to David so he can answer them offline. But David, the
Absolutely.
First question, yes. What are some of the ways we can make sure that we do not overload the autoclave maximum weight limit according to the IFU?
Great question. I usually remember to answer that question while I'm talking about it, but so thank you. Yes, we talked about that because we don't want to overload the autoclave and then create leftover liquid condensate. So a way to do that manually is to weigh your sets. And this is good for twofold. When someone's about to lift a tray up and they see a sign on it that says 25 pounds, you could mentally and physically prepare for that if it's on the shelf. If you think something's going to weigh 5 pounds and you go to pick it up, it's going to kind of really jolt your shoulders. But more importantly, on the back end of that, we would put the burden of that on our autoclave person who was setting up the autoclave loads. They would have a cheat sheet there, and they'd be adding up the weight totals of these sets. For those of us that are luckier to have a tracking system like Census or TDOC or SPM, you could work with those vendors, and you could plug the weights in of every device, and as you're scanning devices to the autoclave truck, it's doing the calculation for you. And then when you hit your weight limit, it won't let you scan anything else to that cycle.
All right, fair enough. Next question is, if you come across a scenario of having to resolve multiple IFUs at my own facility, what should my strategy be? Should I try to resolve it on my own?
No, like I mentioned earlier, and thank you for the question. What I mentioned earlier was that we want to do that risk assessment. We want to get a team together, even if that team, these are trying times right now, even if that team was online, we get an email group going, you get I always like to involve risk. I always like to involve infection control and the operating room, or the end user, the end customer. If it was something in endo, let's involve the endo manager so that we have consensus and it's not just Dave signing off on that item.
Fair enough. And next question is, we once had a scenario where a new device came in, and after we read the IFU, we were not able to reprocess it. Do you have any suggestions on trying to avoid this in the future?
Yes, thank you. And again, that, like I mentioned, if you modify, either there's a capital request process. When you want something that's over a certain dollar amount, there's a process for that. So within that process, there's typically a request form. So whether it's on paper or in the computer, I was suggesting to ask 2 questions, whether it was high-level disinfection required or sterilization. And if one or both or one of those boxes is checked off, it has to kick back to the SPD manager to make sure we have that modality.
Let's see if we can get through one more question. I know we're at the 60-minute mark, but David,
Sure.
To your knowledge, is it a requirement to maintain a paper library of IFUs if you maintain one electronically, both via oneSOURCE and on a SharePoint?
No. You have to have a library. You have to have a resource of IFUs. And the danger of having multiple, I've gone into some facilities where they were printing off of oneSOURCE and putting it into a book. You're just running the risk of doing that. You have to make sure it's updated. You wouldn't want someone to go onto the oneSOURCE and you have a new IFU, and then in your static paper version, or even the version scanned into the hospital computer system, they were not updated. So I would just try to stick to one library. If you have one paper library, then stick to that. If you want to upload it to the computer, then it just lives in the hospital computer. Or if you have the oneSOURCE, use that as your primary source. But it is a requirement. You have to have access to IFUs, and that will be asked by DNV, it'll be asked by Joint Commission, or even the Department of Public Health.